Hi everyone! This is the next episode of MedChem Essentials, scientific seminars devoted to medicinal chemistry. Today we continue talking about metabolism with a focus on phase 2 drug metabolizing enzymes. This talk is a part of a larger whole, and right now we are dealing with topics related to half-life. Presentation will begin with the introduction to phase 2 metabolism, and this will be followed by description of 4 main phase 2 drug metabolizing enzymes. Previously, we described that the process of metabolism is meant to turn a xenobiotic into a readily excitable metabolite through the action of different drug metabolizing enzymes. So we have this lipophilic toxin, and through the phase 1 or phase 2 metabolizing processes, this is being turned to a water-soluble waste being excreted with urine, bile, or stool. The names phase 1 or phase 2, they don't mean that phase 2 always happens after phase 1 drug metabolism. Also, these processes not always produce more water-soluble compounds. There are always exceptions, as with all biological processes. No strict rules. Let's move to phase 2 UDP-glucuronosyltransferases. These enzymes are found primarily in the liver, but also in the kidneys and intestine with 22 human isoforms. As with cytochromes, the share of drug metabolism is not distributed evenly between these isoforms, with some of them taking higher share of the drug metabolism. Up to 70% of clinically used drugs are subjected to glucuronidation, so this is very common metabolizing route. The substrates for UDP-glucuronosyltransferases are nucleophiles. They react with uridine diphosphenoglucuronic acid, substituting this acid and forming highly water-soluble conjugate. Typical substrates are phenols, acids, and other potential nucleophiles. So whenever we have a nucleophilic moiety, we may expect some level of glucuronidation. An interesting fact related to glucuronidation can be found in the case of morphine. Morphine forms morphine-6 glucuronide in the body. This is bioactive molecule, much more toxic than morphine itself. It was identified quite late, because sometime in the 70s, in people being treated for prolonged time with morphine. In those people, this M6G metabolite accumulated to toxic levels in kidneys, leading to kidney failure. This is not the first, not the only example of a case where a metabolite is much more toxic than the parent molecule itself. Now let's move to sulfotransferases. These enzymes are membrane-bound and cytosolic, present in all tissues. They catalyze transfer of sulfonate from a donor to a typically hydroxyl or amine group as an acceptor. Typically, it is, for instance, responsible for the metabolism of endogenous substrate like steroids, catecholamines, or bile acids. When it comes to drug substrates, we can exemplify desvenlafaxine or naloxone, because both of them have phenolic moieties and can undergo sulfonate transfer. Now let's move to glutathione-S-transferases. These enzymes catalyze conjugation of glutathione to electrophilic drug substrates. For instance, we have etacrinic acid. This is a highly electrophilic moiety, and this will get conjugated to glutathione. So here we have the classical example. Glutathione-transferases, they work on par with oxidative metabolizing enzymes. So this is a classical order of the phase 1 metabolizing enzymes catalyzing, like activating the molecule, this happening for the first, and this is followed by phase 2 drug metabolizing enzymes, in this case conjugation with glutathione. Through the action of cytochromes, this molecule, methoxy, will get activated into reactive metabolite, which can conjugate with the glutathione through the active of GST. Why this happens? Because GSH conjugates, they are substrate for various efflux transporters. So our our cells are designed to recognize these JSH conjugates and readily excrete them outside of the cytosol. This aids in the secretion of toxins from cells. These electrophilic reactive metabolites, they may not only conjugate to GSH, but also they can covalently bind to other molecules, for instance, like proteins or any other molecules. And this can lead to very serious acute toxicity, even lethal ones. And these electrophilic reactive metabolites, they are not readily identified in the assays, because in the screening assays, because they are so reactive. So to be able to detect them, researchers are adding glutathione to screening assays to detect these conjugates. If such conjugates are being detected, this is a warning of a possible toxicity. Now let's move to the N-acetyltransferases. We have two isoforms, NAT1 and NAT2. They are expressed throughout the body. They play a major role in biotransformation of carcinogens, cigarette smoke, car exhaust fumes. And self-explanatory, they transfer acetyl moiety from donor, typically it's acetylcoenzyme A, to nitrogen or oxygen function. Drug substrate are anilines, right, procainamide, hydroxylamines, and hydrazines. And this is a very interesting case. Isoniazid is a drug for tuberculosis. It's still being used in practice. It's not a safe molecule. It's quite toxic, which is exemplified by the fact that a single 300 mg tablet for humans is highly toxic to dogs. And this is not related to different expression of these enzymes. It's just the milligram per kilogram dose for dogs, in case of dogs being smaller animals, is much higher. And there were cases of deaths of dogs that by accident ingested these tablets meant for humans. Now let's talk a little bit about the genetic polymorphism. Phase 1 and also phase 2 enzymes, they are subject of genetic polymorphism, and this can lead to toxicity in individual subjects. Interestingly, this isoniazid was found to be differently toxic, to show different safety between humans. Some people showed high tolerance to isoniazid, and other people were highly sensitive to the toxic effects of isoniazid. Upon detailed research, it was found that these people were expressing different isoforms of N-acetyltransferases, which was responsible for different safety and different toxicity of this drug between humans. And so this phenomenon by isoniazid actually polymorphism of enzymes between humans was found as a phenomenon. This genetic polymorphism was also connected to a number of neurological disorders and even cancers. It is a very serious aspect of drug design and development that should be taken into consideration during any drug development. So thank you very much for your attention. That's all for today, and see you next time.